I would start by, again, you've heard some of this before, but really to drive home the point that a significant portion of chronic kidney disease in children, estimated to be 30%, but it may be as high as 70%, results from single gene defects that are inherited as Mendelian traits. In genetic disorders of kidney structure and function, the same phenotype can result from defects in a number of distinct individual genes, thus complicating single gene-based genetic testing. And that was a point that Ashima made. The issue of phenocopies is really an important issue for us as pediatric nephrologists. Gene-based testing can direct targeted therapeutic approaches, and increasingly this is becoming very important. You do genetic testing not just to resolve what the diagnosis is, but actually to help you guide therapy. And I'm going to talk about this in a little bit further detail in a moment. But despite technologic advances, genetic testing is not 100% sensitive in identifying the genetic root cause, that is the mutation detection. And even in cases where the phenotype strongly indicates a specific clinical diagnosis, and this is some of the rationale for having this session today close with a discussion of what happens when genetic testing is negative. Where do we go next? But for us as practicing clinicians, further implementation of genetic testing, really incorporating genetic testing into our diagnostic armamentarium, requires that we have increased genetic literacy. And so I really very much appreciate Hallie taking the time to take us through from basically high school biology all the way to the genetic report that crosses our desk, so that we're really sort of in tune and understand how we should approach genetic testing as pediatric nephrologists, and the importance of approaching genetic testing in partnership with our colleagues that are genetic counselors and also medical geneticists. Technologically, the field really needs to have bioinformatic innovations so that we can do a better job with variant interpretation. I think both Hallie and Dr. Galati talked about the problem of the variant of uncertain significance. Is it real or is it Memorex? Does it really portend that this patient has disease due to this particular sequence change in this gene? Or is it just one of the many variants in their genome that really isn't disease-causing? And for a large percentage of the genetic information that we get back in genetic testing, this is still a conundrum. The large-scale validation of the diagnostic yield for kidney disease, this is more from the perspective of making it more and more likely that our insurance providers will cover genetic testing. Is there a real benefit? Does it advance the care of our patients to include genetic testing? And that figures also into the cost-benefit analysis, both at the level of insurance companies and at a greater societal level. And then finally, and again, this is a point that I think that Ashima made and that I want to punctuate, organization and counseling support in daily practice so that we really know that what we're doing is accurate and is helpful to our patients. I think that's a hugely important challenge for us in busy pediatric nephrology practices. So you've seen this, these data presented in a different way. This is the genomic landscape of pediatric renal disease. So glomerular diseases, there are more than 50 genes that have been identified, everything from congenital nephrotic syndrome to steroid-resistant nephrotic syndrome to Alport syndrome, Fabry disease, Nail-Patella syndrome, Denny's-Drash syndrome, and then, of course, atypical HUS. The tubulopathies, there are more than 50 genes. These can be defects that involve genes that encode transporters, genes that are involved in the development of tubular interstitial disease, as Dr. Bleier will talk about, and then even nephrolithiases. In cystic diseases, there are well over 100 genes, and we'll talk about that in a minute. And then, as Dr. Galati touched on, developmental disorders, there are greater than 50 genes. Most of those genes underpin the congenital anomalies of the kidney and urinary tract, or CACUT spectrum of disorders. But there also are more isolated syndromes, such as BOR, the branchioautorenal syndrome.